Hi guys, today what we're going to look at is blood. This is chapter 12. And this one is in a PDF format. I was having trouble with the PowerPoint, so I had to do that. So you're just going to scroll down on this. The heart pumps blood around the body, so blood is a transporting fluid. It is also a connective tissue. Nutrients are carried in the blood along with oxygen and carbon dioxide, hormones. There's also going to be nutrient molecules that travel in the blood. Very important. Blood also aids in the distribution of heat formed in your active tissues, and it helps to regulate acid-base balance and protect against infection. So we're going to go over the parts of the blood and also functions of those parts. The first thing I want us to look at is if we took blood and to put it in either a test tube or a small capillary tube and spin it in a centrifuge so that the heavier cells go down to the bottom. The erythrocytes are red blood cells here at the bottom. The leukocytes are the white blood cells here in the middle. And also the thrombocytes or the platelets are here in this buffy coat. Plasma then makes up about 50% of your blood. It's mostly water, and it also has some proteins in it. One example of a protein in plasma would be fibrinogen, which is normally in plasma and is necessary for blood clotting. This is made by the liver. Also, albumin is a plasma protein, and this is going to be important for maintaining osmotic pressure within the blood. It's the most abundant of our plasma proteins. Gamma globulins are antibodies, so they're important in helping us destroy certain types of organisms that might come into our body. And then prothrombin is another plasma protein that will help blood coagulate. Coagulation is the same thing as clotting of the blood. Other substances that can be carried in the blood are nutrients, such as glucose. There will also be amino acids and cholesterol. Electrolytes, which are our charged atoms or ions, and these would be substances such as sodium and chloride and potassium. Also hormones, you learned about that in the last chapter. So hormones that are traveling in the blood might be thyroid stimulating hormone on its way from the pituitary gland to the thyroid gland. And then there are metabolic waste products that our kidneys get rid of or might end up in our feces. The term we use for blood cell formation is hematopoiesis. Hematopoiesis occurs in your red bone marrow. There are certain stem cells that divide to form all of your blood cells. Erythropoiesis is what we call the manufacture or the cell division that's needed for red blood cells. So when we have a hormone produced by the kidneys called erythropoietin, and it stimulates these stem cells, we end up with red blood cells. Red blood cells are important because they transport oxygen around our body. So we have to have enough red blood cells to transport oxygen to all of our cells. Red blood cells, when they're in circulation, do not have a nucleus. They originally do, but then they lose that nucleus, and then they live only for about 120 days. And once they get older, their destruction takes place in the spleen or the liver. Now, when the red blood cell is broken down, we end up with one of the substances in that red blood cell that transports the oxygen called hemoglobin. And hemoglobin, once it's broken down, will break into heme and also globin, which is a protein. Heme can be reused and converted into bilirubin, which will be stored in the liver. What I want you to know about hemoglobin is it is a red pigment that's contained inside of the erythrocytes and gives them the red color. And it's made of a protein molecule we call globin. And also in the middle of that, there is a heme group with a central iron. And so iron is very important in the transport of oxygen. Oxygen actually binds to the iron in the middle of the heme. When oxygen binds to hemoglobin, it forms a compound called oxyhemoglobin, which then at the tissues is able to release oxygen to the tissues, and then carbon dioxide is picked up by the cells. So we get that exchange of oxygen and carbon dioxide into the blood. One of the other substances that can bind to hemoglobin is carbon monoxide. And because it actually has a greater affinity for binding to the hemoglobin, carbon monoxide will take the place of where oxygen should bind. And that's why it's so serious when a person has carbon monoxide poisoning. They can't transport oxygen if carbon monoxide is attached to the heme. Hemolysis is the rupture or bursting of red blood cells if a person is given the wrong type of blood. We call that a blood transfusion reaction. But there are other types of diseases that can also cause hemolysis. Another blood cell is a white blood cell, also called a leukocyte, and these do have nuclei. If you look at a blood sample and you see cells that have a nucleus, that's going to be a white blood cell. They can be either granular or agranular. If they're granular leukocytes, they're going to end in phil. So you'll see here neutrophil, eosinophil, and basophil. Those are going to be granular leukocytes. And neutrophils are important because they can phagocytize bacteria. You learned the term phagocytosis back with the cell. That means that they can engulf these inside and then break them down. Eosinophils increase in number in allergic conditions, malaria, parasitic worm infections, and sometimes in people who have bone marrow cancer. Basophils are activated during allergic reactions or inflammation. They produce two substances, one called histamine that causes inflammation, and the other called heparin, which is an anti-clotting or anticoagulant. Here are pictures of these different white blood cells. Notice the basophils have dark stained granules here on their surface, and you can kind of see the nucleus in the background. Eosinophils also have granules, but this time they're red. So these are the red granules, granulocyte, and here's the nucleus. It's kind of a horseshoe shape here. And neutrophils, notice the granules inside are not purple or red, but they're kind of a neutral color. That's where they get their names. And sometimes they're called bands. So neutrophils are going to be white blood cells that have kind of these lobe-shaped nucleus, and these two are actually combined together here. You can't see where they're. White blood cells that are agranulocytes are called this because you can't see granules in the cytoplasm. Another type of white blood cell is an agranulocyte. One of these would be lymphocytes, and lymphocytes can be either B lymphocytes, which make antibodies, or T lymphocytes. So there's different types. The name B lymphocyte comes from where the lymphocyte matures. It's made in the red bone marrow, but it matures also in the red bone marrow. So the B stands for bone marrow. And T lymphocytes are made in the red bone marrow, but mature in the thymus. So that's where T comes from thymus. The second type of agranulocyte is a monocyte. And monocytes can assist in phagocytosis, and they're able to leave the bloodstream and attach themselves to tissues. So they help to isolate the infected area. Because they are phagocytes, they can engulf and get rid of debris or bacteria. So white blood cells can move from the inside of capillaries into neighboring tissues using a process called diapedesis. You can see here erythrocytes, leukocytes, and also platelets. Keep in mind that platelets are not really cells, but only fragments of cells. If living tissue is damaged, white blood cells can cause inflammation. Inflammation occurs when tissues are subjected to some type of trauma. It might be physical trauma, it could be chemical trauma, it could be that organisms get into your body like bacteria or viruses. But the characteristics of inflammation are redness, heat, swelling, and pain. Histamine released from basophils can increase the blood flow into the area and also the permeability of the small blood vessels we call capillaries. Neutrophils quickly move out of the blood vessel to the damaged area by that process we call diapedesis, where they move through the blood vessel, and they can begin to phagocytize bacteria. Pus may also form, and pus is really just dead and living bacteria, dead leukocytes, plasma, tissue. If the damaged area is below the epidermis, sometimes a cavity forms with pus inside, and we call that an abscess. Leukocytosis is the term we used for an elevated white blood cell count, for instance, if a person has an infection, and leukopenia, a decreased white blood cell count. Thrombocytes are very small. You can see the arrow pointing to many thrombocytes in this picture. Thrombocytes are fragments of larger cells called megakaryocytes, and they're going to be used to help in the process of blood clotting. They're important for initiating this process of blood clotting by forming a plug where the blood vessel was damaged. Hemostasis is a process of controlling bleeding and then stopping that bleeding if a blood vessel is severed. So here it's showing the first step of hemostasis. If the vessel is injured, would be vasoconstriction. And what that means is the blood vessel decreases in diameter, kind of pulls in on itself. Also, where the vessel is injured, you'll see collagen fibers that are exposed because of the connective tissue underneath. And platelets start to aggregate there and plug up the area. And then that thromboplastin is produced, and finally a clot forms. And when you look at a clot, it's fibrin threads. So fibrinogen is converted into fibrin, and that is clot formation. Blood type is inherited, and it's due to the presence or absence of antigens on the surface of red blood cells. There are 4 major blood groups: A, B, AB, and O. All of these are named for the antigens on the surface of the cell. So if you know what antigens you have, you know what your blood type is. If you look next to that, you'll see the word antibody. And notice on the first one, if you have A blood, you have B antibodies. You never have antibodies that are the same as the antigen on the surface of the cell. So remember, there's A and B. And so if you have A antigens, then you will have B antibodies. On the other hand, if you have B antigens, then you will have A antibodies. If you have both A and B antigens, then you will not have either antibody. And O blood means that you don't have either one of those 2 antigens. You don't have A antigens and you don't have B antigens, but you do have both A and B antibodies. So in order to know your blood type, you can test for that, and we're going to look at some examples, but obviously you would want to know your blood type before you had surgery or blood transfusions so that the antibodies and antigens don't react with each other. It's going to be the antibodies in your blood, which is mostly what's in your body, that will react with the transfused blood. So for instance, if you have A blood and you were given B blood, you have those B antibodies in your A blood, and those B antibodies would attack the B antigens in the transfusion you were given and cause hemolysis. So that's why we have to be very careful about the type of blood we're given. The universal donor is O negative. Now that gets us to another type of antigen on the surface. Negative means you do not have the antigen we call RH. And so an RH antigen is going to be one that causes a person to have positive blood. If you don't have the RH factor, you have negative blood. If you're RH negative, you still don't have antibodies against the RH antigen. That's just not normal to have unless you've been exposed to somebody that has Rh positive blood. A universal donor is going to be negative then, because they're not going to have Rh factor on the surface that will cause the person's blood to react to it. So that's why that is a universal donor. O negative is a universal donor. If we think about it, if they have type O blood, they won't have A antigens and they won't have B antigens, nothing to react to. And then if they're negative, they don't have the Rh antigen. A universal recipient is going to be AB positive, and that's because they have A and B and they don't have antibodies against those. And obviously positive, and they also don't have antibodies against the Rh factor. Here is blood typing. What you do with blood typing, and this is read as one row here, is you put a drop of blood in each of 3 wells, and then you put a drop of this blue substance in one well, and that's anti-A antibodies. So that is going to combine with any A antigens that are on the surface of this blood. If there's no A antigens, it can't combine. This one, you put a drop of anti-B, which is really yellow in color. You can see it here. But when you put it on the blood, it's going to attach to the B antigen on the surface, if there is one. And then this is a different drop, it's clear, and this one is going to have anti-D or anti-RH antigens, which can then combine with the Rh factor. So if a person has positive blood, then they're going to have this agglutination or clumping if they have that. So let's look at this one. Let's look at this well first. When anti-A is in this well, it caused the cells to agglutinate or clump. That means there is an A antigen on the surface of the red blood cells. In this well, when anti-B was put on it, it didn't clump. So that means that B antigen is not on the surface. And then here we put anti-D or anti-RH on the surface, and it did agglutinate. And what that means then is it does have the D antigen. So if it clumps, it does have the antigen that this antibody is binding to. That means that it does have A antigens and it does have Rh antigens, so it's A positive. The next one, you can see it in this well that you do not have clumping of the cells, but here you do. All this granular is clumping or agglutination. And this one also has clumping or agglutination. So this is B positive. On this one it clumped or agglutinated, and agglutinated. So this is A, B positive. And in this one, this is that universal donor. It doesn't agglutinate, it's O negative. So this is about the Rh factor I was referring to. The other thing you want to look at are normal values for blood. One of the tests that can be done is a hematocrit, which measures the percentage of red blood cells in whole blood. At the very beginning, I showed you a tube that was spun down, and you saw red blood cells on the bottom. In males, you would expect to see 47 plus or minus 5% for a normal hematocrit. So that means from 42%, if you subtract 5 from 47, up to 52%. So if you added 5 to 47, you get 52%. That's normal for men. For women, normal would be 37% up to 47%. For hemoglobin, instead of just looking at the percentage of red blood cells in whole blood, now we want to know how many grams of hemoglobin are there in a certain amount of blood. So normal values for men, 14 to 18 grams. Normal value for women, 12 to 16 grams. Something else I want you to know is anemia, which is deficiency in the number and or percentage of red blood cells and the amount of hemoglobin in the blood. if you think about it, if you have anemia, you're not going to have as many red blood cells or as much hemoglobin to transport oxygen. And this is what leads to the problems with people that have anemia. And obviously there's many different kinds of anemia. Embolism is a condition where an embolus is carried by the bloodstream. And an embolus is a traveling clot. So if we're looking at how blood clots, we expect blood to clot in a broken vessel, but we don't want blood to clot in an unbroken vessel. And if that clot breaks from the wall of the blood vessel and is carried by the bloodstream, it could reach a small artery, like maybe in your heart or in your lungs, and therefore lead to pulmonary embolism or an embolism in your heart, which could cause heart attack. Thrombosis is the formation of a blood clot in a blood vessel. So if we talk about a thrombus, this would be a blood clot that is formed because maybe the blood is traveling too slow and it leads to it clotting. Or if you have changes in blood due to immobility or decrease in mobility. I hope this has helped. If you have any questions, please let me know.